Hello everyone and welcome to our channel Holistic Facts. So in today's video we are going to learn about antibody cocktail since it has been in news for a long time now, and also the first patient that was treated with the cocktail has been discharged and is quite healthy now. So its efficiency is being hailed and it has achieved authorization from USA, European Union, and now in India as well. So, first of all, Roche's antibody was used here, antibody cocktail was used here, right? So, it contains casirivimab and imdevimab. You don't need to go into details of the particular antibodies, but you just need to understand how the mechanism usually works, right? So, since we are calling it a cocktail, it is obvious that it is a combination. It is a combination of these two in a single dose. Then this cocktail is said to reduce the chance of hospitalization by 70% in patients with mild symptoms, mild to moderate symptoms of COVID-19. And then another interesting fact is that the United States President Donald Trump was treated with this last year itself. But the problem here is that it costs 60,000 per dose, which we know is not widely affordable in case of India. But let us see this particular mechanism. Okay. So here, just imagine this particular antigen or pathogen is not bacteria but is your coronavirus, right? We will briefly understand this method because it's not necessary for us to go into molecular details, right? So the pathogen enters your body, macrophages attack these pathogens, right? And they disintegrate the particular pathogen, process the fragments, and they show those fragments on their surfaces, right? So these exposed fragments, T cells and B cells bind to such fragments, study these fragments, and they produce antibodies against such fragments so that they can bind to oncoming pathogens and create an immune response and inactivate those pathogens, right? So this is basically how antibody formation works. Now we need to just simply remember that T cells and B cells are associated with such kind of immunity. And whenever we talk about long-term memory while producing antibodies, it is associated with the B cells because this long-term response. See, T cells also create memory cells, right? But in vaccine associated responses, it is mainly memory B cells. Then another important fact here is that we need to understand what is polyclonal antibody, right? So whenever naturally we get certain infection in our body disintegrates this particular oncoming pathogen into various fragments which each can act as an antigen, and for these different antigens different antibodies are created by various B cells and T cells, right? And this is a mix of many antibodies, and hence we call them polyclonal antibodies, right? So now that we have understood what is polyclonal antibodies, let us see what is a monoclonal antibody. So first of all, a monoclonal antibody is responsible for only one type of antigen, right? So let us understand this now. So if this is your pathogen, in this case it is coronavirus. So whenever we talk about polyclonal antibody, it is going to be a mix wherein various antibodies will recognize various antigens. For example, some antibodies might recognize this part, some might recognize this part as foreign, some might recognize this part as foreign and attack the pathogen. But in case of monoclonal antibodies, remember, these are produced artificially in laboratories, isolated, and each type of antibodies that is produced from a particular type of cell is isolated. This particular type of cell is then multiplied so that the oncoming clones of this particular cells will produce only one type of antibody which will be responsible for only one antigen, right? So let's say, for example, for only this particular part. So these are pure antibodies and not mix, and hence these are called as monoclonal antibodies because they are clones of single cells, produced from clones of single cells. So what is the advantage of monoclonal antibody, right? Because it sounds like polyclonal antibodies might be more efficient since they recognize many parts of the same pathogen. So the advantage is that monoclonal antibodies, because they are artificially created, they are created in lab, and they are highly reproducible. Large scale production is possible and unlimited production is possible here. And whenever you require, You can just produce them. You don't need to go through entire process of again infecting an animal, isolating the cells, separating the cells, and then producing antibodies. No. Once you have already perfected that method, you don't need to even maintain the animals, right? And that is why this is advantageous. Other than that, the speed and sensitivity and specificity assays are quite efficient when monoclonal antibodies are used, and hence in research these are used. But sometimes it can be used in treatment as well. So now this is just another concept here. I think that you need to know that antibodies can work in two ways. It can simply block the entry of virus into the cells that are usually affected. So here an example of COVID itself is taken, right? So SARS-CoV-2 virus attaches to ACE receptor through its spike protein. So antibodies can simply block that spike protein. Then another thing that can happen is that many antibodies can actually simply coat the virus, and then this virus and antibody or antigen antibody complex can be recognized by macrophage and can be engulfed and destroyed, right? Why I have given this concept here is that sometimes UPSC asks questions which are quite indirect, in which only concepts can help you have the answers, right? Then there's another concept which is very important, and I think there can be questions about this time, because there's a lot of talk about antibody therapy, right? The convalescent plasma therapy was also in news, although now it is discontinued. So there's active immunity and there's passive immunity. So active immunity is developed due to production of antibodies in one's own body. So for example, if I am infected with some kind of pathogen naturally, my body will recognize it as foreign and will create antibodies and fight it off, right? So my body has participated actively. But in case of passive immunity, maybe in some patients it is not possible to form antibodies in a scale that is required to fight off a particular infection. In that case, the antibodies are produced artificially in laboratories and then injected into the patient's body. So this is known as passive immunity, because the patient is passive in this case, right? Patient's body is passive and the antibodies have come from outside. So this is given here because it is important for us to understand that antibody cocktail that we are talking about is a type of passive immunity. Then who should take the antibody cocktail? So it is to be administered for treatment of patients with mild to moderate symptoms, right, in adults, plus who are at high risk of developing severe COVID-19. Now we know that if you have some kind of underlying conditions, comorbidities, you are already at higher risk of severe COVID-19. So if such a person gets infected with COVID-19, he or she can be administered with the cocktail within 48 to 72 hours, and then it can be very effective. Then in pediatric patients, that is children above 12, can be given this who weigh at least 40 kg. And this therapy is not recommended if patients are already hospitalized and require oxygen support. That means if the COVID-19 has already progressed to a severe level. Then another thing here to understand is that Regeneron and Roche antibody cocktail have received emergency use authorization in USA, European Union, and in India, right? But under what provisions has this been given? So in India, there is no such thing as emergency use approval in Indian rules, right? We talk about accelerated approval process in our 2019 rules, wherein we approve vaccines or drugs. So in what cases such accelerated approval can be given? If a drug is still in clinical trial, but it's showing meaningful therapeutic benefit, or if a drug is intended for treatment of a very serious or life-threatening disease, or a disease of special relevance to the country, which COVID-19 is right now, right? And also to address unmet medical needs. Then again, in case of new drug or a vaccine that can be considered for approval for remarkable effectiveness is reported even from phase 2 trials, right? So in phase 2 itself, if remarkable effectiveness is seen, it can be given authorization. But in such cases, additional post licensure studies may be required. So even after giving approval, the constant data collection and studying the effect in the population of such drugs is required. Then the approval granted is obviously temporary, because this is emergency use. This is for emergency use. This is accelerated approval process. You're not going through traditional ways, right? You're shortening the time period for approval. So this is for temporary reason. So this is something that you need to keep in mind. and many new things are constantly going to come up. And remember that in science and technology section specifically, uh, biological things that are related to antigen antibody responses or our immunity are quite of importance for upcoming prelims examination. And so I hope you liked today's video, and if you did, please like, share and subscribe to our channel Holistic Facts. Thank you.